Hi, my name is Nathan Treff. I'm going to be lecturing today about comprehensive chromosome screening in a human embryo. I'm currently the Director of Molecular Biology Research at Reproductive Medicine Associates of New Jersey, which is one of the largest infertility clinics in New Jersey, and we consistently have among the highest success rates in the United States. I'm also an Associate Professor at Rutgers Robert Wood Johnson Medical School. Just some notes. Please feel free to submit any questions during the lecture by typing them in, and I'll do my best to answer those questions at the end of the talk. So, in vitro fertilization has been around for a while. Dr. Edwards and Steptoe first performed IVF and developed the first baby, Louise Brown, many years ago, and he recently won the Nobel Prize for that activity. Dr. Edwards also pioneered a number of other things, including preimplantation genetic diagnosis, where embryos can be genetically tested after culture in a petri dish and before transfer back into the uterus. And actually, the most common genetic abnormality that we currently test for in human embryos is for the presence of aneuploidy. Aneuploidy is essentially the wrong number of chromosomes where you have too many or too few, and it increases with advancing maternal age. We also know that the prevalence of infertility significantly increases with advancing maternal age, which is shown here in this graph, where you can see in orange the continuous decline in the success of IVF after transfer back into the same person's uterus. If you were to take donor eggs from younger women and transfer back to those same patients, even of advanced maternal age, you then get success rates which are reversed back to what you see in younger individuals. So we know that one of the primary causes of infertility is associated with advancing maternal age, and we also know that aneuploidy significantly increases with advancing maternal age. So as a result of that relative inefficiency in the success of having a child with advancing maternal age, that we can actually successfully treat infertility with in vitro fertilization, and we bypass a lot of the problems, but it's still a relatively inefficient process, despite the fact that it is the most effective treatment of infertility. And as a result, we end up having to transfer back more than one embryo at a time in order to achieve a relatively good success rate with IVF. And so you can see in this graph that the percentage of embryo transfers, which over time involved more than one embryo. And in 2010, only 19% of the embryo transfers were actually a single embryo transfer. And one of the problems with this is we have multiple gestations, twins and triplets. Approximately half of IVF deliveries are twin deliveries or multiple gestation deliveries. It's because we transfer more than one embryo back in order to achieve decent success rates. And what you see in this graph is that with twin and triplet pregnancies, there is a much higher prevalence or percentage of births that are preterm and also low birth weight infants. So there's a lot of complications associated with IVF, and this is the primary complication, multiple gestation, and it's because we transfer more than one embryo. So essentially, we'd like to improve the treatment of infertility by better selecting embryos, and we think that one way to do that is to select embryos that have the right number of chromosomes by first screening for the presence of aneuploidy within the embryos prior to selection for embryo transfer. Unfortunately, previous attempts, which primarily involved the use of what's called fluorescence in situ hybridization, really failed to show the expected benefit. And many different organizations came out saying, in fact, that there's no evidence that what was originally termed preimplantation genetic screening, or PGS, actually improved outcomes. One of the statements was from the American Society for Reproductive Medicine, and they actually did at least suggest that it wasn't successful the way in which it was currently performed, and that really opened up the opportunity to develop newer technologies which are more comprehensive than fluorescence in situ hybridization. There are many limitations of FISH, one of which is that, in fact, it doesn't look at all 24 chromosomes in human cells. It only was able to look at about 12. So you ended up transferring embryos that had abnormalities for the untested chromosomes. And now, for about 7 years, newer technologies have been developed. Actually, metaphase CGH has been around a little bit longer. We also have single nucleotide polymorphism or SNP array-based methodologies. Another version of CGH involving arrays rather than metaphase spreads, and this is actually a little more commonly used now. There have also been qPCR-based methodologies, and now there are many labs developing next-gen sequencing capability, again, all to look at all 24 chromosomes in the embryo. So I'm just going to briefly talk about some of the basics behind a few of these methods before I get into the details of how they've been validated to actually demonstrate the expected benefit. Now, the majority of these methods are only possible because of a methodology called whole genome amplification, because when you have DNA from the human embryo, there's very little material to begin with, so you really have to amplify it in order to test across the whole genome. And there are a couple of different methods of whole genome amplification. What I'm going to show you here is a PCR-based strategy, which begins with DNA intact. It's then fragmented, and then an adapter is ligated, which then serves as a primer site, which you can then do PCR to amplify all of those fragments. And then there's a variety of methodologies you can use to characterize that whole genome amplified DNA. As I mentioned, SNP arrays, array CGH, and also now some developing methods for next-gen sequencing. With one particular type of SNP array, what you have is your embryonic DNA, whole genome amplified, and then labeled. It's hybridized to the array. You then obtain DNA from known normal samples and process those independently. And so those normal samples are also labeled the same way, but hybridized to different arrays. And then you can computationally compare the data from your embryo to the data obtained on other arrays from known normal samples. And what you obtain is a relative copy number for all the chromosomes, as shown here with this copy number plot, where you have 0, 1, 2, 3, 4, or 5 copies, and then each of the chromosomes. And this is an example of a cell that came from a sample with trisomy 20, and that was male. So you see an extra copy of chromosome 20 and a missing copy of X. Because it's male, there's only one X chromosome. Array CGH is a little bit different. So again, though, you start with whole genome amplification, and you label your embryonic DNA with one dye. You then do the same process on known normal DNA, but label it with a different dye. You then mix the samples, and they compete for hybridization to the array. So what happens is, if you have, for example, more DNA for a particular chromosome in your embryo, then that particular dye will be more intense and actually look more green, for example, when there's a gain. And in contrast, if there's a loss, so your embryo has less DNA than the normal sample, it will look more red. And these signals can then be interpreted again to look at copy number across all the chromosomes. So you can see that with the arrow where there was a chromosome with a gain or a trisomy. With next-gen sequencing, obviously it's similar in terms of obtaining sequence information, but it's massively parallel. So you obtain many reads for a particular region in the genome. After you obtain all these individual sequence reads, you then align it to the reference genome. And in this example, there were 14 reads which aligned to this part of the genome. And essentially what you can do is count the number of reads which align to each chromosome found in the cell, and use those relative counts to determine the copy number of each chromosome. And some groups have used whole genome amplification again by doing sequencing on the embryo, counting the reads that you get on each chromosome, and then comparing similar process on normal DNA, and the amount of reads on chromosomes in those samples are compared computationally to your embryo. Now, this particular methodology is still under development, unlike array aCGH and SNP arrays, which are currently clinically utilized. But there are many groups, like I said, developing this methodology, so we're likely to see more of this in the future. Another methodology which, unlike what I've talked about previously, involves quantitative real-time PCR. So there's no whole genome amplification step. The embryo biopsy is put into a PCR tube, lysed, and then a multiplex PCR reaction is performed. And this really is a focused PCR instead of whole genome amplification. And it's much faster, it's less expensive, and much more flexible than whole genome amplification in terms of the various applications for preimplantation genetic diagnosis. Once you've done the multiplex PCR, the samples are loaded onto a 384 well plate, and then real-time PCR is performed so that you look at individual positions in the genome individually in independent reactions. The methodology has been described in fertility and sterility. You can actually, with this methodology, unlike the others, obtain results within 4 hours, and this really opens up a lot of possibilities in the clinic because we often need to get results in a short turnaround so that, for example, we don't have to cryopreserve the embryos in order to wait for the answer and then decide which embryos to transfer. This has also been applied not only to look at aneuploidy, but also single gene disorders, which are relatively common, and one of the reasons people go through IVF is so they can have unaffected children. We also can look at translocations and mitochondrial DNA mutations. Interestingly, we've also been able to apply this to look at microdeletions. And this is an example of what some of the raw data looks like. Here we're looking at a sample that we knew possessed trisomy 16 and trisomy 21. And what you see, it's basically a heat map showing the copy number for all 24 chromosomes. In the upper left, there are 16 data points within that rectangle for chromosome 1. And then it goes all the way down to X and Y. We use a relative quantitation method that's previously been described, but with a few modifications to that methodology. Essentially, again, we get these copy numbers, and what I've highlighted here are areas which you can see there's increased copy number, and this is actually chromosome 16 and 21 as we would expect. And we also see a decreased copy number for X and Y because this is from a male sample. And when you plot the copy numbers across the chromosomes, this is similar to what it might look like. And you can see, again, a gain of 16 and 21 and a loss of X and Y. And this happens to be a sample that we use routinely for quality control to make sure that the assay is working over time. Now, I mentioned some of the limitations of fluorescence in situ hybridization, and one of them was that there really was no good validation prior to routine clinical implementation. So we've spent a lot of time really validating this particular type of screening before routine clinical application, and we refer to it as comprehensive chromosome screening, or CCS. One of the first things we did was to look at the preclinical accuracy, and that involved looking at consistency within cell lines which had previously well-characterized karyotypes, as well as consistency within embryos. We also wanted to investigate the safety of the procedure. One of the reasons why previous methods may not have worked well is because there may have actually been a risk of performing an embryo biopsy. So we looked at both the predictive value, which I'll talk about, the impact of biopsy to address the safety of the procedure, and then, of course, some randomized controlled trials to look at the clinical efficacy, again, before routine clinical implementation. This is an example of data with the SNP array methodology, just illustrating how you can use cell lines with, again, previously well-characterized aneuploidies to establish the validity of your test. The difficulty here is being able to get an accurate diagnosis from a single cell. On the left are results for material where you have a large quantity of DNA from these cell lines, And again, you get the expected karyotypes, which are typed in there, including a variety of trisomies. And then single cells from those same cell lines were obtained and evaluated. And when we do this in a blinded manner, we did actually demonstrate 98.6% accuracy on single cell 24 chromosome karyotyping. A similar study was performed with qPCR, again on positive controls, and here we had approximately 98% accuracy. As I mentioned, it's also important to look at consistency within the embryo because it is possible that the assays won't perform the same on an actual embryo as they do on cell lines, which are an entirely different cell type. And in this particular study, one of the things we tried to control for was the known presence of mosaicism within the embryo. And it actually can make it difficult to interpret results when you get more than one diagnosis from the same embryo. It's not clear whether your assay itself failed or whether there was real variation within the embryo itself. We know that mosaicism exists, and it happens because you can have chromosome errors occur during division after meiosis and during mitosis, so that you would have some cells, for example, with an aneuploidy, and maybe some other cells within the same embryo that are euploid. So depending on which part of the embryo you biopsy, you might get one result. And then if you rebiopsied another area of the embryo, you could get a different result. So to try to control for that when testing the validity of an assay, we really focused here on embryos which had two consistent diagnoses from two different biopsies using SNP array technology. We then rebiopsied those same embryos and evaluated them for consistency using qPCR. Again, to try to eliminate the biological variation from assessing the variation of the assay itself. And here we showed approximately 99% consistency between qPCR and SNP array technology. And this was also important because showing the equivalence of qPCR to SNP array actually allowed us to benefit from some of the clinical trials that we had done using SNP arrays to justify the use of qPCR in future studies. We also went back and evaluated whether or not the particular methodology was consistent within the embryo by looking at a comparison with fish-based methodology. Here it also illustrates one of the other limitations of fish, which is really an inconsistent diagnosis of single cells. So here we took cleavage stage embryos, which we then biopsied out individual cells for aneuploidy screening. Those individual cells were randomized so that approximately half were evaluated by fish and half were evaluated by SNP array. And I mentioned the issue of mosaicism, but in this design, the level of mosaicism should be randomized and equivalent in both groups. And actually, when you look at the results, the fish-based methodology failed to give a consistent diagnosis in any of the embryos assessed, and actually would have predicted 100% mosaicism. And that was significantly higher than the level of consistency we saw with microarray, where we would have only predicted a 31% mosaicism rate. So this really illustrated that FISH is an inconsistent methodology, and it actually leads to a clinical problem because many programs would perform a biopsy at the cleavage stage at day 3, perform FISH, but then obtain a blastocyst from that embryo, which looked very nice under the microscope and otherwise would have been considered for transfer. But because the day 3 FISH came back with an abnormal diagnosis, that embryo would normally be discarded. But if the FISH actually overdiagnosed aneuploidy, you would actually be discarding an embryo with potential for the ability to make a baby. So the question is whether you actually discard that embryo or biopsy, rebiopsy and cryopreserve the embryo for additional analysis. And we actually did that in about 50 embryos. We obtained multiple biopsies after those embryos had been diagnosed as aneuploid on day 3 by FISH. And what we found was that in 4 different sections of the same embryo, actually all 24 chromosomes were euploid, despite a day 3 FISH diagnosis of aneuploidy. And it really indicates that FISH really poorly predicts blastocyst aneuploidy, and it likely resulted in erroneous disposal or otherwise reproductively competent embryos in a large majority of the cases. And this goes to the negative predictive value of the test. You really have to address whether or not your test can accurately make an abnormal diagnosis and whether it actually predicts a negative clinical outcome. To do that, one study we performed with the SNP array-based methodology was what we call a non-selection study. So the embryos were selected. Here you see a green and a blue embryo, selected based on morphology. And that's how we normally select embryos in the clinic. But before transfer, we biopsied the embryos and saved those biopsies for future analysis. They did undergo SNP array-based aneuploidy screening, and we also used the SNP data to fingerprint. So any embryos that implanted and we could obtain DNA from the conceptus, we then compared to figure out which of the embryos actually made the baby. We could then go back and ask whether or not our SNP array-based screening methodology successfully predicted the actual clinical outcome. And one of the most important measures we obtained from this was the negative predictive value. That is, how often we would have predicted through aneuploidy screening a negative clinical outcome, failure to sustain implantation. And the predictive value of this particular assay was extremely high, and such that we could actually say we could safely use the methodology to discard an embryo, unlike what likely occurred with fish, where there was probably a significant overdiagnosis of aneuploidy. We also, so that data is shown on the right, the two bars on the right. The white bar indicates the negative predictive value when looking at blastomeres, and the black bar indicates the negative predictive value of looking at a trophectoderm biopsy, both of which were high. So we could successfully predict the negative outcome. And we also looked at the positive predictive value, which turned out to be much better when the assay was performed looking at the Blastocyst stage embryo, so later in development. Taking a trophectoderm biopsy was more predictive of a positive outcome. When we predicted euploidy from a trophectoderm biopsy, those embryos sustained implantation more often than if we had a euploid blastomere from the embryo. And so there are a variety of methodologies. One method that's being used quite a bit in the clinic today is aCGH. However, very little validation, level 1 evidence actually exists to demonstrate validity of aCGH in diagnosing aneuploidy in the embryo. We collaborated with a group in Italy to evaluate embryos that had been diagnosed as aneuploid by aCGH. We looked at about 55 embryos. Those embryos were rebiopsied and then blinded before performing qPCR-based comprehensive chromosome screening. We did obtain the same result in 87% of those embryos, but 13% gave a different diagnosis. We then took those 7 embryos and again rebiopsied and performed blinded SNP array-based comprehensive chromosome screening. We found 2 embryos in which all 3 methods didn't match, and this is actually evidence that those particular embryos were truly mosaic. But there were 5 embryos in which 2 of the 3 methods matched. In all 5 cases, the SNP array matched the qPCR, and therefore the aCGH was interpreted as having provided an error. And in all cases, it was a false positive aneuploidy diagnosis. Here's an example where you see the aCGH result predicting a monosomy 8. And then qPCR and SNP array in 2 different laboratories performed on the same embryo in a blinded fashion showed prediction of euploidy in both cases, again indicating a false positive aneuploidy prediction by aCGH. And again, of course, 0 cases where the SNP and the aCGH matched, indicating a significantly smaller error rate from aCGH, from qPCR compared to aCGH. So I've talked a little bit about some preclinical validation, looking at safety by evaluating the actual negative predictive value for clinical outcomes. Another important question is when is it best to apply comprehensive chromosome screening? I briefly mentioned you can do this testing at both the cleavage and the blastocyst stage. And it turns out that this question hasn't really been well addressed, even though embryo biopsy has been performed for over 20 years to prevent genetic disease. We do think that it is important to perform biopsies in order to directly evaluate the genetics of the embryo for a lot of different indications. It may actually be useful to improve IVF outcomes, which I'll talk about when we get to randomized controlled trials. But again, there's no definitive studies on whether embryo biopsy hinders implantation or what stage is optimal. The majority of the world really, up until more recently, performs testing at the cleavage stage by obtaining an individual blastomere as opposed to a trophectoderm. Again, though, it's really important to evaluate these methodologies and understand what might be the safest. There are many theoretical advantages to doing a trophectoderm biopsy over a blastomere biopsy. One of the studies we completed was a paired randomized controlled trial. Okay, and here we again applied DNA fingerprinting. 2 embryos were selected based on morphology, so the best looking embryos under the microscope. They were then randomized so that one had an embryo biopsy performed and the other served as a control and was not biopsied. We then transferred both embryos prior to analysis, and then any singletons which occurred, we then performed DNA fingerprinting to determine if it was the biopsied or non-biopsied embryo. And then over a number of patients, how often was it one or the other? And we did this by looking at both, here what's diagrammed is the cleavage stage blastomere biopsy, but we also evaluated the impact of trophectoderm biopsy. This summarizes the results of that study, where we're looking at the sustained implantation rate, and now this is essentially the delivery rates. On the left are the results after cleavage stage biopsy, and you can see that there's a significant reduction in the reproductive potential of embryos that had a blastomere biopsy performed, shown in red, compared to the non-biopsied controls. In contrast, when you look at a blastocyst biopsy where you obtain trophectoderm, you can see that there was no significant difference in the reproductive competence of a biopsied and non-biopsied embryo, again in this paired randomized control trial. As I mentioned, there's a lot of theoretical advantages. When you remove a blastomere from a cleavage stage embryo, it represents a larger percentage, about 13% of the volume of the embryo. In contrast, when you do a trophectoderm biopsy, it's probably less than 5% of the mass of the embryo. In addition, we really don't know when we take a blastomere whether or not that particular cell was destined to become part of the inner cell mass, the actual embryo itself, or the trophectoderm lineage, which would give rise to the placenta. And also, it's actually technically challenging. It doesn't necessarily impact the reproductive potential to consider this, but the accuracy of diagnosis of a single cell is not as good as when you have multiple cells. So there's some practical considerations there as well. When you perform a trophectoderm biopsy, in contrast, it's after embryonic genome activation. Again, it's a smaller percentage of the embryo. And it also represents an extra embryonic biopsy. So we're not actually taking any cells from the inner cell mass, which is destined to become a fetus. So with all those theoretical considerations, we actually perform this randomized trial and support those theoretical estimates with actual evidence that blastomere biopsy is really not safe and blastocyst biopsy is. So this has led us now to exclusively perform blastocyst biopsy when performing any type of genetic testing on human embryos. Another important set of experiments are randomized controlled trials to actually demonstrate the expected clinical benefit in the way in which the assay will be used clinically. The first randomized trial that was performed was with qPCR-based comprehensive chromosome screening, and we demonstrated an increased implantation rate per embryo transfer that was, in fact, significant. almost 20% improvement when using CCS compared to patients randomized to the control group. We also see an increased delivery rate per embryo transfer and increased delivery rates per treatment cycle, which is typically what most people are interested in. So with the use of comprehensive chromosome screening, we do see an improved delivery rate. So it does, in fact, give the expected clinical benefit when using this particular methodology of comprehensive chromosome screening. And I mentioned that one of the main complications with IVF is multiple gestation. We started hypothesizing that better, and other people have suggested that better selection of embryos would actually improve the success of single embryo transfer, which we know would reduce significantly the level of multiple gestation after IVF. A retrospective study shown here indicated that single embryo transfers without CCS, shown in blue, had lower success rates than those embryos which had undergone CCS and were considered euploid. And that was true in all age groups. This led us to perform a second randomized controlled trial, which was a non-inferiority randomized trial, where we wanted to establish that, in fact, we could get equivalent success rates with a single euploid embryo compared to the standard of care, which is transferring two unscreened embryos. And what you see here are the success rates in blue of the cases where CCS was performed and a single euploid embryo was transferred, compared to the control where two untested embryos were transferred. So we did see equivalent ongoing pregnancy rates, which hadn't been shown in any randomized trial looking at single embryo transfer. And then one of the secondary measures was looking at the risk of having a twin gestation. And here you see that with a single euploid Blastocyst transfer, 100% of the pregnancies, as might be expected, were singletons, whereas almost 50% of the two untested blast transfer cases in the control group were actually twins. So there was a significant reduction in the prevalence of twins with the use of a single euploid embryo transfer with CCS. So comprehensive chromosome screening has shown that to be accurate using an analysis of controls with known aneuploidies for some particular methodologies, again, unlike fluorescence in situ hybridization. It's been shown to be consistent within embryos, looking at blinded analysis and against other technologies. I mentioned that because of the PCR-based nature of the assay, you can actually test for single gene disorders, translocations, as well as insertions and deletions in parallel. And all of this can be performed within a 4-hour turnaround time, which is really critical in many cases of PGD for IVF. We've shown that it's safe to perform through Blastocyst biopsy in a randomized controlled trial, and that it's effective in improving IVF success rates. We've also shown that it's effective for elective single embryo transfer, essentially eliminating twins but maintaining the success rates as transferring two untested embryos. And this methodology really is now much more widely applied. Even in our own clinic in the last 5 years, we've seen an increase from 5% of our patients to almost 70% of our patients using this technology to improve their outcomes. And we think this has also led to an overall improvement in IVF success rates at our program. Future directions, I mentioned the incorporation of next-generation sequencing. We're going to see a lot more of this applied to human embryos during IVF and PGD. We have recently published a paper involving the use of NGS, next-gen sequencing, for single gene disorders. And I mentioned there's going to be a few talks probably at the American Society for Human Genetics and the American Society for Reproductive Medicine meetings. One of the benefits of having this capability is we can now start looking for other markers of reproductive potential. we still see cases where embryos predicted euploid fail to make a baby, and so we think that there are other potential markers to better select embryos, as well as test for the synchrony between the endometrium and the embryo itself. What we at least can do is control for aneuploidy within these research studies. So by eliminating aneuploidy from the equation, we can then focus on what may be predictive within euploid embryos. And there are many ongoing technologies such as time-lapse photography or morphokinetics, proteomics, and metabolomics. There was recently some press around this time-lapse imaging of embryos and the potential use of those videos to predict whether or not embryos are chromosomally normal. Unfortunately, it's really not that predictive. Embryos that were predicted to have low levels of aneuploidy still had 36% aneuploidy. In addition, the embryos with predicted to be less euploid still had 31% prevalence of euploid embryos. So we don't necessarily think that this type of analysis will be useful for chromosome predictions or aneuploidy predictions, but within euploid embryos, it may help to further refine our ability to select reproductively competent embryos. And there are some populations of patients which we're still not sure will benefit from CCS, and so we have some ongoing trials to investigate subsets of IVF patients, which, again, we're not sure whether or not there is a benefit yet for those particular people. And with that, I'd like to thank the organizers of this meeting, all the members of my laboratory, RMA of New Jersey, various funding and collaborators. And I'll go to the Q&A. Yeah, so are there any questions? I'd be happy to answer. Yeah, thank you. Sure. So anyone interested in getting the CME credits can go to this link, I believe, and follow the instructions. That's it. Okay, thank you.